To everybody who has joined, welcome and hello. And this is a very special meeting that was just came about in the last couple of weeks. For those who don't know, my name is Mike McMorris. I'm with Livestock Research and Innovation Corporation. Our focus is the future of livestock production, and today's topic is highly relevant to that. During the presentation, if you have questions, which I'm sure you will, please hold them to the end and put your hand up in the chat function at the bottom, and Jean will then know to unmute you and ask you to ask your question. Alternatively, if you prefer, simply put a note in the chat and Chloe will track those. So I have the great pleasure of introducing Michael, who's going to give the presentation today. Dr. Rogers is a Tier 2 Canada Chair in Food Nanotechnology at the University of Guelph. He studies the underlying mechanisms that facilitate small molecule self-assembled nanostructures and their behavior along the alimentary tract. Dr. Rogers' work is highly recognized, and he has held faculty positions at Rutgers University and the University of Saskatchewan before joining Guelph in 2015. I first met Michael when he made a proposal for research funds looking at the digestion of plant-based meat versus real meat. I was intrigued then, and I'm excited now that he is sharing what I believe to be some very important results. So with that, over to you, Michael.
Thanks, Mike. Good afternoon, everyone. So today I'll be giving the talk, There's More to Food Than Just Composition. And in this, we'll be comparing A&W's Beyond Burger to the hamburger, from the bolus to the stool. So from the time of swallow to the time of excretion, using some of the most advanced simulated digestive equipment available. This project was run by mainly Zitong Zhu, who's a PhD student in our lab. And this was really a collaborative effort between myself and Dr. Lapointe, who runs the Twin Shine, which we'll get to. So when we talk about food today and the food environment we currently live in, lots of things have changed. And one of the most startling facts that I found is, for the first time in human history, non-communicable diseases, cancer, diabetes, cardiovascular disease, all of which have a part related to diet, is responsible for a larger percentage of mortality than communicable diseases. Why isn't it moving? Let's try that again. There we go. And it's these increase in diet-related diseases has led to the United Nations referring to it as a public health emergency in slow motion. And then COVID hit. And COVID kind of threw everything for a loop. But still today, in Western society, the single largest cause of mortality and morbidity are diet-related chronic diseases. And 50 to 65% of the adult population are afflicted with some sort of diet-related disease. And the challenge with this is that diet-related diseases don't stem from a single element of consumption. Instead, it's the complex interactions of those foods over time. And in the past few generations, what we have seen is the development of an entirely new epidemic that was unheard of a century prior. And this is metabolic syndrome. And every time a developed nation introduces ultra-processed foods, we see the population's incidence and occurrence of metabolic syndrome drastically increase. And if you're not familiar with metabolic syndrome, obesity is fundamental to metabolic syndrome, as that precedes the emergence of impaired insulin sensitivity, dyslipidemia, so high cholesterol, and hypertension, all that correlating to a 1.6-fold increase in mortality. So what has caused such a large subset of the population to be diagnosed with metabolic syndrome at such a young age? Well, if we look at our history from the mastery of fire, which is really the beginning of human evolution, when you talk about the occurrence of Homo sapiens. So after the Neolithic Revolution, we saw availability of food change, which allowed us to form settlements, domesticate crops, but it didn't really change the foods we were consuming. Through the second agricultural revolution, same thing, we increased food production, but didn't really change the fundamental diet of society. And this still held true as we hit the Green Revolution. But at the same time as the Green Revolution, the Industrial Revolution was occurring. And as the Industrial Revolution was occurring, we were at the same time really learning about nutrition, because if you think about the Nobel Prizes of classical nutrition, they've only started since 1900. So our understanding of human nutrition is still really in its infancy. And the Industrial Revolution has changed today's food environment through the mechanization and industrial scale production of highly processed, shelf-stable foods. So if you look at the two pictures below, the pre-industrial revolution food environment was almost all whole foods, whereas today, the food environment consists mainly of boxed, formulated, ready-to-eat convenient foods. And the FAO, the Food and Agricultural Organization of the United Nations, has recognized a disturbing trend of displacement of dietary patterns based on meals and dishes that are made from unprocessed or minimally processed foods. And those are being substituted increasingly based on ultra-processed foods. So if you're not familiar what an ultra-processed food is, we use a NOVA classification of foods, which came out about 5 or 6 years ago. And really what they're looking at isn't the composition of the food itself. Instead, it's the history of processing that that food has undergone. So group 1 foods are minimally or unprocessed foods. This is edible parts of plants, animals, so meat, also fungi, algae, and water. Unit operations here don't really alter the molecular level of the food. Instead, it's more of a process that is applied to preserve it without altering. So freezing is a good example of a type 1 food. Type 2 are processed culinary ingredients. They're typically used in type 4 foods. So they are things like ketchup, mayonnaise, and those are key in formulating the ultra-processed foods. Now, processed foods are foods that have undergone a fairly severe process, but are still recognizable as a group 1 food. So a cured meat would be a good example of a processed food. Now, group 4 foods are the foods that we refer to as ultra high processing. These foods are formulated from ingredients that are extracted from whole foods. And the foods do not resemble the whole foods that they were derived from. This can include hydrogenation, extrusion, molding, certain frying applications. Key here is hyperpalatable, low cost, convenient, and long shelf life. So we are seeing consumers slowly migrating from group 1 foods and more relying on group 4 foods. And why is that bad? Well, we know intrinsically that eating a cob of corn is not equivalent to eating a box of corn pops. So when you think about the food or agriculture in Canada, when we, at time of harvest, only about 5% of the sweet corn actually goes to the sweet corn market, kiblet. So frozen corn, canned corn, corn on the cob. 45% goes to silage, and about 45% of that goes to formulating ultra-processed foods. So corn can be refined and separated into starch, protein, which would be cornmeal, oil, it can be flour, and then we can also convert it to various corn syrups. With these ingredients, we can formulate entirely new foods. Those foods do not resemble the whole foods that they were contained from. So eating a cob of corn is not the same of eating deep fried corn chips. And believe it or not, the average Canadian consumes 48% of their calories today from ultra-processed formulated foods. Now, these tend to be higher in salt, sugar, and saturated fats, but there's more than just composition that's changing in these systems. So our objective was to compare how meat digests versus a plant-based burger. Are they the same? Do they digest similarly? Do they release the fat in a similar way? We know carbohydrate digestion is distinctly different, as Beyond Burgers contain carbohydrates and beef doesn't, minus the bun. The second question is, even consuming Beyond Burgers infrequently, can it change the microbiota of our colon? So in doing so, we use two of the most advanced systems in the world, the TIM-1, which simulates the upper GI, stomach, duodenum, jejunum, and ileum, while Objective 2, looking at the microbial outcome, relies on the SHIME, which simulates the large colon. So let's start with the TIM digestion. So the TIM is a computer-controlled, automated digestive system. It simulates every aspect of digestion from the mouth to the end of the duodenum, at which point that food would transition to the colon. So our meals were standardized to feed the exact same amount of fat, because that's really what we were interested in. What was the lipemic response, or the rate of fatty acid release during digestion? And to do this, we had to simulate for 6 hours, and every 30 minutes, we imaged and measured the lipid bioaccessibility. Now, the neat thing about the TIM-1 is it is compartmentalized into 4 main units. The stomach, which simulates the low pH, temperature, as well as it controls the secretion of enzymes and acids, controls temperature, controls the mixing via peristalsis. So actually what we see is a contraction between those two chambers. So it's not like a stir bar, it's actually like the antral wave contraction of your stomach. Then through the dynamic 6-hour digestion, the meal slowly transits from the stomach to the duodenum, following the gastric emptying rate of a healthy human adult. Once it reaches the duodenum, the system controls the pH, regulates it back up to around pH 7 through bicarbonate secretion. It also secretes fresh bile, which we acquire from Conestoga meats during pork slaughter, and pancreatic enzymes. Once the meal reaches the duodenum and ileum, that's where we begin to take out the digested fractions, which you can see the 2 membrane filters here and here, which are the colloidal filters that allow the fatty acids and monoglycerides to pass through the colloidal membrane. So what we're simulating is the luminal conditions up to the point of absorption. Now, when we talk about food science and the ability to change, let's say, a glycemic response of a food, it's not that we're changing the rate it absorbs, we're changing the rate that it digests within the luminal fluids. So this is termed the bioaccessibility of a macronutrient. The bioaccessibility refers to the available nutrient that's available at the surface, ready for absorption. This is different from bioavailability, which is the amount of the material that is available to perform the physiological function. So this bioavailability also includes absorption, circulation, and first pass metabolism. Bioavailability does not account for that. However, bioavailability is often the bottleneck of digestion because the enzymes are the rate limiting step. So the rate of digestion and the rate of monomeric substrates that can cross the epithelial is the bioaccessibility, and that's what we're looking at in this system. So within the TIM, we had already done a few years prior looking at different degrees of cookedness on how muscle is digested. In this study, we've included some of these results for comparison sakes. So here we have raw steak. We also have a steak that has been cooked to the extreme, so 85 degrees Celsius. And then we have our beef burger and our plant burger. We can see that in the stomach, after 30 minutes, there is significant myofibril structures remaining in the meat products, whereas even after just 30 minutes in the stomach, the structure remaining for the plant-based burger are very, very small particulates. And if you think about the fact that digestive enzymes have to work at the interface of the food matrix or the chyme, this then changes the rate lipid digestion will take place, because plant burgers increase in surface area through digestion and early on occurs much more rapidly than in beef. If we move to the duodenum, you can see that in cooked beef and in the hamburgers, you can see residual structures persisting up to and including 240 minutes into digestion. Whereas again, when we look at the plant burger, we see that the majority of the structure has been reduced to very, very small particles. This is again exaggerated in the ileum. And after 300 minutes, again, you can still see there's undigested meat particles moving through the tim, while in the plant-based burger, they are all but small, dense, spherical particles that have been highly digested. So what does that mean? Well, there's two things that are important. First, simply looking at the changes in microstructure don't give us the entire picture for lipid digestion, because although the structure of the food determines how that enzyme can interact with the lipids, it's also dependent on where the lipids are in that food. So if you look at the top micrograph, beef burger versus Beyond Burger, you can see that the beef burger's oil, which is stained pink with Nile red, is confined very closely to the dense particle structures, which would make sense because the fat is deposited along the myofibril fibers. Now, the Beyond Burger, which is formulated, you can expect that the oil doesn't have as strong association with the food matrix, which is very clear in S30 for the Beyond Burger, where you can see a really diffuse pink scatter. So that means the lipids are not as associated with the solid structures in the Beyond Burger as they are with beef. So as that structure disintegrates, you can see very quickly at J120 that the pink is very quickly being dispersed in the Beyond. However, in the beef burger in the duodenum, you can still find dense locals where there's a lot of undigested lipid. Even at 240 minutes, if we compare, the Beyond Burger has a very, very fine dispersion of lipids. And again, the meat still has it associated with the structures. So this shows us the lipids are rapidly coming out of the network during digestion for the Beyond Burger. So what does that mean when we talk about lipidemia? So a lot of people know and understand the glycemic index of foods based on sugar. However, just as important to the glycemic response is the lipemic response. And the fasted free fatty acid blood titers are becoming a very important marker for health. So how fast do the lipids digest in Beyond versus a hamburger? So in the first three graphs, ABC, this is the non-cumulative digestion that happens in every 30-minute interval. So you can see at 180 minutes in A in the duodenum, the Beyond Burger, the plant-based burger, is digesting at a much, much faster rate than the hamburger. And what's interesting there is irrespective of the type of meat we looked at, so a whole steak that's either raw or cooked, they digest very, very similar to a hamburger, which really makes sense because when you mince meat, although we're breaking up the scale above the micron length, below the micron length, we don't have much effect when we make a hamburger patty. So when we look at the cumulative response and the total cumulative bioaccessibility, figure F, we can see the rate of digestion for the Beyond Burger is significantly higher than for the three other samples: cooked meat, raw meat, and the hamburger. If we want to present this in a little bit different view, this is the final bioaccessibility after 6.5 hours. This shows you that the plant burger, the lipemic response associated with eating a plant burger would be 3 times greater than eating meat. That would be about the same as eating a piece of bread versus a highly digestible chocolate bar, where the bread would be more similar to beef and the chocolate bar would be more similar to the plant-based burger. So there is clearly a very big difference in the bioaccessibility and rate of digestion of the lipids. Again, this would also have a much higher glycemic index because whole meat contains very little, if none, carbohydrate, whereas the Beyond Burger contains significant amounts of sugar, of starch. So what's interesting is not only would it have a higher glycemic response, but it would also have a much higher, well, we show has a much higher lipemic response, which is not desirable. So steady and slow is the best way to think of digestion. You don't want a big influx of macronutrients inundating the system. When that happens with sugar, we get an exaggerated insulin response, and over time, this reduces the sensitivity of our the receptors on our muscles that actually allow that sugar to be uptaken and utilized. So after we found out the glycemic response was so different, the lipemic response, we went and looked at how does the human microbiota change? And this experiment is a very long experiment. So how it works is we have two systems running in parallel. We take both systems and we inoculate it with a fecal colony obtained from a healthy adult male, and we allow that to stabilize in the Scheim system. Typically, once we've added the fecal matter, it takes 4 weeks for that bacteria to reach the stabilization. And how do we quantify that? Well, two ways. One is we make sure the population isn't changing by looking at the 16 RNA sequence, and we look at the metabolites being produced by that bacterial population. Once we've reached a stabilization of 3 days where it has produced the same amount of metabolites and the same population, we can say that the stabilization of those two sets of containers is now in parallel. So when we start the experiment at the treatment point, we have two setups. We have one setup that's going to run the Beyond Burger and one setup that's simultaneously going to run the hamburger. So these two sets of conditions are mimicked starting with identical microbial populations. So we have these two chambers that are ready to receive the meal that are identical in composition and activity. What we then did is we fed the system, we substituted the basal growth medium and supplemented either the Beyond Burger or the beef burger. So one set of treatments was fed a hamburger every day, and the other set of treatments was fed a Beyond Burger every day. Following that week, what we did is we went back to the basal growth medium and looked again at the metabolites in the population to see if changes that occurred during treatment were maintained during washout, or did the population return to what it was during the stabilization period. We know that this tends to be the case and is the challenge for probiotics in foods, is while consuming the probiotic, you would be in the treatment state and your gut microbiota responds to that treatment. However, upon conclusion of taking probiotics, those populations typically slowly move back to what they were initially. So when we're doing the stabilization, key to that is having the different pH ranges of the three regions of the colon. So coming from the ileum, we enter the ascending colon, which is the first region, typically maintains a pH of about 5.6. Then it moves to the transverse colon, where the pH is increased, and finally the descending colon, where the pH is increased again. Another key difference between these three chambers is in the ascending colon, it's receiving the nutrients from that meal. That then metabolizes and produces a series of metabolites that then go to the transverse colon and can actually be referred to as metabolic cross-feeding, where the microbiota that consume the food in the ascending colon produce metabolites that the bacteria in the transverse and descending colon can utilize. And we see this. So here is our microbial community composition. There are many, many species of bacteria present in both Beyond and the hamburger's meal. Looking at just the population, it's very difficult in A, B, and C to discern if there's any differences between the samples. So there was a couple of techniques we could do to see what, if any, was the difference. And the first thing we wanted to look at is what is the ratio of firmicutes to bacteroidetes? Why were we interested in that? Well, this was one of the first findings of the gut microbiome that actually correlated with obesity, where they find if you have more fumicutes, you have a tendency to have higher weight. And what was interesting was that in the beef supplementation, bacteroidetes were far, far more abundant, whereas when fed the Beyond Burger, the undesirable firmicutes saw an increase. So what we actually saw in this result, and really all this is the comparison of A, the blue bars to the red bars. So the blue bars are the firmicutes and the red bars are the bacteroidetes. And what we saw was for beef, they favored a lower FB ratio, which does not associate with obesity. The Beyond Burger did associate with a larger FB ratio and could be implemented in obesity if it has permanent changes. Next, what we did is called a Lefse analysis. And what that does is it compares the change in population across all of these different bars. And that gives us D and E. So in the AC, early on in the colon, we see the bacteroidetes are enriched. So that's the red bar. And for the Beyond Burger, the blue bars, Clostridiaceae and Plantaceae are enriched. If we move over to the far column, this is the combined result from both the ascending and descending colon. Sorry, the ascending and transcending colon. Something really interesting happened in the descending colon, but it was unfortunately difficult to assign a reason for. But in the Beyond Burger, we didn't get enough DNA to quantify the microbial population during that region of time. So there could even be greater negative implications associated with the Beyond Burger as we weren't able to acquire the DNA in sufficient quantities from those samples. What's interesting, though, is twofold. One is the Beyond Burger saw an increase in Citrobacter, and that's not good. Citrobacter is a pro-inflammatory gram-negative bacteria associated with irritable bowel syndrome and gastroenteritis. So the upregulation of Citrobacter for the Beyond Burger is definitely a detriment and was not observed upon consuming beef. Now, when you consume beef, we saw a major increase in 5 species. All 5 species confer health benefits to the host. So Blatia produces short-chain beneficial short-chain fatty acids, essential in intestinal homeostasis and permeability. Megasphaeria protects against diarrhea. Rickenselia reduces visceral obesity. An abundance of all stripes is negatively correlated to obesity. So the population changes is much more favorable upon consumption of beef compared to the Beyond Burger. Now, if we look at the metabolites that those microorganisms produce, there's some really interesting things that we notice. First, by T7, so this is treatment day 7, this is where our effect is going to be the greatest, we see in the ascending colon a greater proportion of all short-chain fatty acids. Why? Because saccharolytic bacteria are using the starch from the Beyond Burger to produce short-chain fatty acids. So this was actually an expected result. And when we remove the Beyond Burger and go back to the basal medium, we see that effect is washed out. Now, when we go to the transcending colon, we have to remember that the nutrients reaching this have been already exposed to the bacteria in the ascending colon, partially metabolized, and the metabolites produced from the AC have entered the TC. So the bacteria production of short-chain fatty acids in the TC is less of an effect of the food that is present in the lumen, but of the metabolites now that come from the AC, or the ascending colon. And what we see is a drastic increase in the short chain fatty acids, again, a desirable trait in the transcending colon for the beef burger. So now what the assumption is that the beef burger improves the microbial population that can utilize protein for energy, and that utilization then produces the metabolites which enter the TC and have actually formed a synergistic effect with the microorganisms that are present later on in the colon. This effect is seen less again when we move to the DC, but it still persists. And what's really interesting and probably most surprising is for butyrate, that elevated butyrate production, again, which is a very, very desirable trait, remains after a week of washout with basal media. So once you've stopped consuming that hamburger or Beyond Burger, you can see that the production of metabolites starts to return in the ascending colon. You almost see the same metabolite production in washout day 7 as standardization. But when we get to the descending colon, we have actually changed the population, and the metabolites they're producing remain higher even after the exposure of the beef burger. So this suggests a couple things. First, short chain fatty acid producing bacteria, Megasphaeria, was upregulated in the beef consumption, and acetate and propanoate producers, Bacteroidetes, Alstrips, and Blatia, were also upregulated. This shows the importance of metabolic cross-feeding between microbial species in different regions of the gastrointestinal tract. Now, what's interesting is switching back to the basal growth medium, what we term washout, did diminish the effects of the concentration of individual and total short-chain fatty acids, except for butyrate. And that remained significantly higher for beef burgers beyond the washout, suggesting that these changes may persist beyond the treatment effect time. So what does this tell us? Well, they can argue that plant-based burgers may be organoleptically similar. They may have similar micronutrient compositions, but they do not digest the same as meat. And because they don't digest the same as meat, they are not nutritionally equivalent. Differences in the nutritional composition and nutrient abundance between beef and plant burger alter the gut microbial profile, or the populations of bacteria, and the metabolites they produce, the short chain fatty acids, which have a symbiotic relationship with the host. Yet, composition fails to account for the distinctly different physical structures. And those distinctly different structures alter nutrient degradation and ultimately macronutrient digestion and bioavailability, as studied in the TIM. This shows that although we have mastered the dissociative techniques required to create ingredients from whole foods, our ability to take those ingredients and reconstruct healthy foods at this point is very limited. And with that, I'd like to thank you for your attention, and I would be happy to take any questions.
Wow, great. Thanks, Michael. I'll start with a question. So when we went through this the first time, you showed us results, and it was in essence in real meat, the fats are bound to the structure of the muscle, whereas it's not in the plant-based. At that time, I was thinking about cellular production of meat, which my understanding, correct me if I'm wrong, is basically a big vat and you produce the muscle, but then to make it desirable to consumers, you have to add the fat, which I'm assuming is then not bound to the structure of the meat. So would you expect the same results from a cellularly produced hamburger?
Likely. So I'm actually a part of a Genome Canada grant that's going in this semester for that. And it's interesting because we aren't even close on that front from a food application. We haven't even identified the suitable strains of muscle cells to use in whole cell growth. So absolutely, you will not be able to configure the lipid configuration the same way the biochemistry of an animal does it. So yes, we would see similar effects with whole cultured meat as we would with formulated ultra-processed foods.
Thanks. And we have a question here from Sylviane. Are other protein sources such as tofu and other vegetable-based sausages and patties, which are also ultra-processed, equally poor nutritionally and bioaccessible as plant-based burgers, which are marketed as meat replacers? How does their composition compare?
Yes, great question. And this for me really touches home to dairy products as well. We see a lot of attempts to try to mimic the organoleptic properties, but the results will be the same. They do not digest similarly.
So having if somebody wanted a plant-based burger because they just like the taste, it's kind of safe to say it's kind of like, okay, have it as a treat, but it's kind of like eating a potato chip.
Absolutely. 100%. Yes, all formulated foods behave the same. So if you're eating a can of Campbell's soup, it is not as nutritious as if you produce a soup at home with all fresh ingredients, limited salt, limited loss of food structure, limited number of times it's been exposed to thermal processing. All of those steps each degrade the nutritional quality slightly. So you think in tofu and any of these products, the more individual steps that are taken to produce the food, the more rapidly typically it digests. Now, with something like tofu, it's a little bit challenging because tofu doesn't have a whole food equivalent. There wouldn't be something that you could say, this is the counter argument to that. In another study we did, we compared ultra-processed formulated Pringles chips, which takes potato starch, potato flakes, and mixes them together and fries them, versus a sliced potato. And again, the lipemic response of the sliced potato was drastically lower than the ultra-processed Pringles. So the Pringle would fall under a class 4 food, whereas the sliced potato would fall under a class 3 food, which is a processed food. But when you look at that potato chip, you can still recognize to some extent it came from a potato.
Mm-hmm. Other questions? I could keep going with questions, but I'm sure some must have some. I see no hands. This is more of a philosophical question. So one of the big issues facing the livestock sector is the possible replacement of meat and other animal products with plant-based cellular fermentation, fill in the blank. There'll be other technologies. Those technologies are raising a ton of money, like unbelievable sums of money. They haven't supplanted livestock products yet, maybe more on the milk side than certainly on the meat side. If you were the national minister of agriculture and there was a brief before you debating whether or not these products should be allowed to be called meat and milk, what would your opinion be?
No, it's absolute. There should be brand identity to Whole Foods. We should not. We see it with ice cream. You know, there's some brand recognition that it's got to contain dairy in ice cream, but for meat, to put meat on the Beyond Burger, to me, is entirely misleading, and it gives consumers a false comparison to the nutritional value of that product, which is very, very dangerous. The other thing too that we have to remember is livestock rearing in Canada does not use the same land required to grow peas. So even if we supplanted the meat industry, the land utilized for raising cattle would not go to growing crops. When I went out to Alberta and looked at the pasture lands, you could never terraform those to an extent where you could cultivate large scale crops. So that's one thing we always have to bear in mind is we have some bad land in Canada that we can't grow crops, but we can raise cattle on. That's good. That's good use of land. So when we talk about carbon footprints and all that, there's other aspects we have to take into consider. And I think the thing that I would give all advice to anyone is eat as whole as you can. Avoid boxes as much as possible. And again, if you think about how our digestive system has evolved since the onset of fire, when we introduced fire, we can actually see in populations today that if you are in a northern climate, your length of ancestry with use of fire was much longer than near the equator. And that has translated to a loss in dentation. So people that are from Scandinavia have smaller tooth sizes than people from South Africa or South America. And that's to do with the softening of foods during digestion or during cooking. So if cooking can cause our teeth to change, think about what taking away all food structure does to our digestive system. So the enzymes, the activity of those enzymes, the ability of us to absorb nutrients, all of that has been closely underwent evolutionary pressure for the last 10, 20, 30,000 years. And now we have such a neocentric change in our food supply that some adults consume almost no whole foods anymore. And that's extremely dangerous and extremely unhealthy.
It's an interesting challenge for the livestock sector because people in general are pretty self-centered and short-sighted.
Yeah.
And so this is really getting at much more of the detail around people's food than they actually understand or get into or care about, frankly. They
Yep.
tend to care largely about price. I mentioned all the money that's going into cellular production. A lot of that is driven by investors looking for return on their investment. At the end of the day, though, from a human health perspective, this may be an unfair question, but any advice you have to the livestock sector on how to better get across its message that whole is good, meat is good, in the think of the bigger picture beyond immediate gratification of taste or stockholder investment?
Yeah. And I think my messaging would be we cannot create a two-class food system in Canada, which we are rapidly doing. And what I mean by that is if you look at the socioeconomic status of neighborhoods, what happens is we get a disproportionate number of fast food chains, convenience stores in low income neighborhoods. And those tend to have less grocery stores, things like Whole Food stores. So in those low income neighborhoods, let's pretend we have a single parent with one child, no car, working a minimum wage job. If you prepare dinner that night, are you going to buy a $5 head of broccoli? Or are you going to go to McDonald's and buy a Happy Meal? We have set up our food distribution supply chain to really focus on delivering ultra processed foods to the most number of consumers. And some consumers don't even have access to whole foods if they wanted to buy them. So what I would say is if you are concerned about price, eat ultra processed. If you're concerned about longevity, you need to consume the foods that our ancestors consumed, which are whole foods, low in refined ingredients, and low in added salt and sugar.
This is going back a little bit, but my mind's been wandering as you've been speaking about. We tend to focus more on the meat. We've talked a little bit about other replacement animal, quote unquote, products. So things like almond milk and oat milk and all the variety of other milks that are out there. How would they, what would you expect them to compare to milk versus going through your system?
Yeah. And again, light and day difference. So a few years ago, we actually compared infant formula to breast milk. And the results, again, in that were staggering. Breast milk digested much, much more slowly than infant formula. So when you talk about satiety, how hungry you are again afterwards, if that food rapidly degrades and falls apart, your hunger drive kicks in sooner. You eat a meal again. That leads to obesity. This is integrated all the way from that. You can actually see differences in the gut bacteria of breastfed versus bottle fed. So there's changes there. So milk, we cannot mimic. There is no dairy equivalent that mimics the casein micelle. And the casein micelle is what makes dairy extraordinarily good for you. So without the casein micelle, you don't get the added calcium. You don't get the enhanced calcium bioavailability, solubility. Fatty acid profiles can be fairly similar from the milks, depending on what source it is. But one of the big key differences is the fat content. So in animal-derived products, typically the fat is in stearic acid. When we start looking at the palm oil derivatives, almond milk, we start to get to the medium chain triglycerides. The evidence is starting to shift for saturated fats, that all saturated fats are not bad for us. And if that's the case, that would give another argument that why animal products are better for you. If stearic acid is heart neutral, where palmitic oleic are not, that would be another reason why the Beyond Burger and formulated foods would be unhealthy. So again, when you make dairy milk when you make a milk substitute, there is nothing that mimics the casein micelle. And why that's important is when the casein micelle digests, again, it releases a lot of calcium, but that hairy layer of protein that stabilizes it is very specifically digested. And what happens is it goes from milk very quickly into a cheese in your stomach. The solidification of that means transit time is slower, means gastric emptying is slower, means your bioaccessibility of lipids takes longer, means you have better control of the absorptive process of dairy products compared to the milk. So I would imagine lipids would digest the same, but you would lose the effect of the casein micelle to carry calcium, and you would lose all the other stuff, ceramides, sphingolipids, lactoferrin system, all these other bioactives that are so important in milk and have very beneficial health outcomes. Those aren't emulated in dairy mimics, and they just don't exist. So I think there would be differences, but I think they would be different than what we see here, but underlying same messaging. Your dairy milk is likely much, much better for you.
I've got one more question. It relates to the studies that looked at breast milk versus milk replacer, and it had an impact on the microbiome.
Not biome, digestibility. We didn't do the biome with that one.
Oh, sorry. Okay.
But there has been studies that have compared. So there's the major establishment of the gut microbiome is dependent on four things. If you were born vaginally or C-section, has a lifelong altering effect. If you're breastfed or bottle fed, if you have antibiotic use, and I forget what the fourth one was, but it's something about socioeconomic status.
So would you expect the microbiome of a 50-year-old that was breastfed to be different than a microbiome of a 50-year-old who was bottle fed?
Yep.
Fascinating.
Yeah, and then that literature exists already.
Yeah. We have a question here from Wayne Hiltz with Manitoba Chicken Producers. How do we get Health Canada to be aware of this information, and how do we get CFIA labeling regulations to acknowledge these concerns?
Yeah, I don't have a good answer to that. Yeah, that's out of my wheelhouse. I honestly think that it's going to be very difficult to mandate it through government. I think we need a grassroots movement. I think the general population of Canada has become too far removed from its food production, and they don't know really conceptually what food should be. And the foods that make up your diet are so important. So if they don't understand the difference between a whole food and an ultra processed food, government regulations won't alter necessarily their decision to consume. What we need to do is make sure that consumers are well aware that the time they spend in meal preparation is worthwhile. And if they want their children to have a longer life expectancy than them, they have to move to a whole food diet. Ultra processed foods are going to limit our life expectancy. That will be the major determinant in my generation.
Wow. And Wayne's question, I think, is excellent because we seem to be finding having to do messaging now when the counter messaging is that cows are the new coal, and that you hear people who are very focused on a single issue say we just need to we need to stop farming.
Yeah.
I mean, if you're involved in agriculture, that seems laughable, but I mean, that's the messaging that's out there. So we have a huge challenge ahead of us. But this is, I think, the reason I was so excited when you showed me these results is that there's a really, really good message here. We have to figure out how to get it across, but it is a really good message for the livestock sector.
Oh, it's great.
I don't see any other questions. We're running up on the hour. There is a recording of this. We will be sending the link out to Michael. Thank you so much for your time. I find this stuff really exciting, and hopefully we can make good use of it, and all the best in the future. Thanks again.
Thank you. And if anyone online has any questions, my email's at the bottom. Feel free to send me an email. I'm always checking it, always on it. So I'm always happy to respond.
You need to balance your life more if you're always on email.
I know, I do. I really do. My wife tells me all the time.
But it's appreciated. Great. Thanks, everybody. Have a great day.
Thank you. Enjoy the rest of your day.